Okay, let's move to viruses. So, looking at kind of trying to put things into perspective here, let's look at the size of viruses. They are little tiny because here's our cell, here's the nucleus, here's an animal cell. This is the size of a bacteria, zero point 0.5 micrometers. I mean, this is like so incredibly small. And then the virus is even smaller than that. So we're talking about something very small. Wendell Stanley, back in 1935, he was the first person to actually isolate the virus and a virus, and the virus was the tobacco mosaic virus, or we call it TMV. So this tobacco mosaic virus is making these leaves turn yellow here. Let's look at the structure, the actual, what does a virus look like? So if we're looking at the viral genome, the virus possesses either a double or single strand of DNA or RNA. And this is how viruses are actually identified, is by whether or not they have a single or double strand. Viruses can carry they do carry very small amounts of DNA. Most are only 4 to 500 genes total, so they carry very small amounts. And they have these structures, this viral protein coat, and it's referred to as the capsid. And so it has this capsid, and you can see the RNA is inside here. The capsid actually serves two purposes. One, it protects the DNA or the RNA that's inside of here. And it's also used as the attachment of the virus to the host cell. So you can see here's that capsid in there, and it's used as the attachment point. It's built from protein units called capsomeres, and so these individual proteins are capsomeres. You can see them here, and you can see inside here is the DNA. And some viruses can also have this viral envelope, and basically it's a cloak derived from the previous host cell plasma membrane. So it is an example of mimicry. It's a great example, honestly, of mimicry. It looks like a normal cell, but it actually is like a Trojan horse because the danger is inside of it. The AIDS HIV virus has a viral envelope that's derived from the T helper white blood cells, and we're going to look at that in a minute. We have a picture of that. All right. Now, let's see. So here's another picture. Here's the RNA inside. Here's the capsid around it, and then if it has an envelope, here's the envelope. And so it attaches, you can see it's attaching to the host cell's plasma membrane. It attaches, it injects this capsid with the DNA or RNA inside of it, and then this becomes the viral genome, and that comes in, it serves as a template, and it starts replicating inside of that host. And then once it replicates, it's going to burst out of that host cell, and now it's created new viruses, multiple new viruses as a result. Now here's a picture of a bacteriophage, and it attacks bacteria. We also call them phages, and it's a virus that attacks bacteria. So here is a bacteria, E. coli. Here's the outer membrane of it. And you can see, here's the DNA that's inside of the capsid. And it's going to insert, that DNA, that viral DNA, into that bacteria. And the bacteriophages, those are the largest and the most complex of the viruses. Now, I want to point out that viruses are not living organisms. They cannot be killed. They can be broken apart using chemicals, though, but they can't be killed. That's why viruses recirculate throughout the body. And we just looked at, in the last video, we looked at the characteristics of living things. And while the genetic material is copied from the virus, it does not meet the characteristics of being alive. And so we do not consider bacteria alive. So let's look at some viral reproduction of what happens. The virus must have a host cell in order to reproduce. Now, they're considered what's called obligate intracellular parasites. That's a sweet sounding word, isn't it? Man, I'll tell you, you throw that out at a party and you are going to be cool forever. Or not, but so obligate intracellular parasites. Parasites, of course, reckon it harms its host while it benefits. And so the virus, as the name intracellular indicates, the virus has to get inside the host cell in order to reproduce. Now, the viruses need to use that host cell, the ribosomes and the enzymes, to make either the DNA or the RNA strands and the new capsomeres to form new viruses. We also have something called the host range, and that host range refers to, think about it, a host range. This is a whole range of what organisms a virus can attack. So they don't just attack anything. There's specific viruses that have a range of what they can attack. And it's determined by its recognition of certain glycoproteins or the glycolipids that are on the host cell membrane. So the virus can recognize those proteins or those lipids that are on there, and they're attracted to that. All right, let's go back up here. So there are these restriction enzymes. If you remember when we talked about restriction enzymes before, those are the cutting enzymes. So the restriction enzymes that are found in the bacteria act as a primitive defense against the viruses. So the enzymes cut up the viral genome and then inactivate the genes from being transcribed. And they're called restriction enzymes because they only cut at a certain nucleotide sequence. In other words, they're restricted to what they can cut. So they can cut up that virus. All right. So when we talk about something called a retrovirus, when we say something so retro, it means it goes back. Now, this retrovirus is a unique type of virus. Retro means to reverse or go backwards. And what they use, a retrovirus, uses reverse transcriptase, ACE. So reverse transcriptase, meaning it's an enzyme. And what it does is it turns RNA into DNA. So instead of DNA and transcription going from DNA to RNA, reverse transcriptase does the opposite. Okay, so here's your reverse transcriptase. We're going from RNA into DNA. And so it's turning messenger RNA into that double-stranded DNA. So then it can be incorporated into the host DNA. So AIDS and HIV and the common cold viruses are all of those are both of those are retroviruses. Okay, so you can see this process of how that works. So reverse transcriptase, messenger RNA into the DNA. Let's go back up here. So the major viruses, HIV, AIDS, Ebola, influenza, SARS, those are major ones. When we call something an epidemic, that means there's a big outbreak in one area. If something's a pandemic, this is a global outbreak. Just think, pan is going to be that whole global versus the epidemic. So if there was an epidemic in Birmingham, if there was an outbreak of something in Birmingham, we'd call it an epidemic. But if it was globally, then we would say it's a pandemic. The plant viruses, there's over 2,000 that are known to exist, and this is big. These plant viruses, we think of animal viruses affecting us, but plant viruses are huge because they totally affect a farmer's crops and will destroy those crops. Now the viroids, so viroids themselves are naked infectious RNA molecules. So they can attack plants only. Oid means like, and they are like viruses. So viroids are not actual viruses, but they're like viruses, and they're going to attack the plants. Prions, these are infectious proteins. And things like mad cow disease is one example. And mad cow destroys brain cells, thus driving the cow mad until driving it mad until it dies. The human version is the KJD. So Creutzfeldt-Jakob disease is what we call in humans. In animals and obviously in cows, it's called mad cow disease. But this is how it affects it. If you look here, here's the prion, here's a normal protein. It affects it and transforms it. And you can see now, look what we have. And then the ones around it will then be affected as well. Oh, that's all we have. Bye-bye.